Bulletproof Radio, a state of high performance.
Hey, it's Dave Asprey with Bulletproof Radio. Today's cool fact of the day is that most people talk about measuring ketones, these fat-burning molecules, in the urine. You pee on these little test strips. But did you know that ketones can be detected just by smell? The simplest ketone is called acetone. It's actually a nail polish remover. And it leaves through the lungs and can give you like a sweet, fruity smelling breath. And if you stay in ketosis for long enough, the primary ketone will not show the same intensity in your urine, but you can definitely measure it with your breath. In fact, I have a ketone breath meter somewhere in my stack of toys, and I imagine today's guest probably has more of them than I do. But before we get on to today's guest, I'm a little bit of a coffee snob. I love trying new coffee, but I learned a few years ago that I don't like how I feel when I try new coffee because there are extensive problems in coffee with mold toxins and other things that just simply aren't bulletproof. So I enjoy the taste, but I don't like the after effects. That's why I developed the original Bulletproof process to create Bulletproof. And we developed a medium roast profile that has a ton of flavor. But I wanted a little bit more variety in my coffee. So we've been working for the last year with one of the top coffee tasters on earth, Paul Songer, who trains the judges for the Cup of Excellence, which is like the world championship of coffee. And I am so pleased to announce 2 new roasts of Bulletproof processed coffee beans. These are mold-free, independently lab-tested coffees. And they're made at our Bulletproof plantations. We just changed how we roast them. The first new roast is called The Mentalist. It's available in whole bean and ground. It's a medium to dark roast, kind of right in the middle there. It's got dark cocoa and vanilla aromatics that open up to a rich, full-bodied coffee with some cherry sweetness and notes of almond and caramel in there as well. It's profoundly good. I've been putting it in my espresso machine, and man, it's crazy. You take it, and it's engineered to taste amazing with butter and brain octane oil. A lot of coffee tastes bitter and weird when you put butter in it, not this stuff because, well, we built it that way. The next one is for you if you really like your dark flavored, dark roast coffee. It's called French Kick. It's a dark roast, but it's not charcoal. It's smooth and sweet. It's pleasantly, almost a little bit smoky, and it's got baking chocolate notes, but it finishes really clean and has a medium body. This is one of the few dark roast coffees I like. I am not a fan of those burned things that you might find at the corner coffee shop. This isn't one of those, but we pushed it as close to dark as we could get and still maintain that flavor for you. You will be amazed at how you feel when you have this coffee, just like you are with our existing coffee. And now you've got a whole palette of coffees you can play with, depending on if you like espresso, French press, immersion, or just drip brewing. However you like your coffee, you can do it, and you're going to just be amazed at the flavors. I'm excited to share these with you. Today's guest is someone who's been on the show before. In fact, several years ago, he was somewhere around episode 80 guest, and it's none other than Dr. Dominic D'Agostino. He's an associate professor at Morsani College of Medicine at the University of South Florida, traveling senior research scholar at the Florida Institute for Human and Machine Cognition, a neuropharmacologist, and he's probably best known as one of the top ketogenic diet researchers out there, specifically looking at neurological diseases, cancer, and other chronic illnesses. Certainly one of the biohackers I respect the most. Dominic, welcome to the show.
Thanks for having me, Dave.
I wanted to have you back on the show because, well, it's been too long and because we had such a fun time on the first show. And we'll put a link in the show notes so people can go back and hear the first interview, which was, I think, one of the very early ones you were giving about ketosis in the overall public sphere, as far as I know. And I learned a lot on that one, realized we had some similar recipe ideas around chocolate and huge amounts of fats and how it could make you feel amazing. Before we jump into the main reason, which is I had Dr. Veach on the show recently, and Dr. Veach is a major researcher in ketones as well. And he had some really interesting comments, some things I had not heard anything about in the keto community. So we're going to get into that stuff. But for people listening who aren't super ketogenic already, let's talk about just ketogenic diets in general, do a little bit of a primer catch-up from one of the top researchers in the field here. And if you're a longtime listener, you probably already know what ketosis is, but we are now getting sometimes a little bit more than 2 million downloads a month on Bulletproof Radio. So there are lots of new listeners who are just getting into this. So we're going to go a little bit basic on ketosis, and then we're going to go deep on, okay, what kind of supplements should you be using? What kind of diet should you be using? And we'll just have lots of fun because we always do. All right. So what are the top benefits of a ketogenic diet that you've come across in your research? You know stuff about this that most people don't. So what is it, just the 2-sentence version, and then what's it doing for people that you're seeing in the labs?
Well, the history of the ketogenic diet is really interesting. We wrote a 3-part article, actually, with Travis Christofferson. It might be good to have on your show. And it's on Rob Wolf's podcast. And it's the history of the ketogenic diet, part 1, 2, and 3. So if your listeners want to check that out. But I got interested in the ketogenic diet because it mimics the metabolic physiology of fasting, right? And we knew for centuries, if not millennia, that fasting is very effective for controlling seizures when drugs fail. In millennia, 1,000 years ago, they didn't have drugs or more. So the diet was designed sort of to mimic the physiological effects of fasting and prevent seizures. So that was the original application, was specifically for seizures that result from oxygen toxicity that are experienced by the Navy SEALs using a closed circuit rebreather. So that work has expanded to a broad range of other seizure disorders, including things like glucose transporter type 1 deficiency syndrome, Angelman syndrome. We have other models of seizures we're looking at, neurodegenerative diseases, including Alzheimer's disease, traumatic brain injury, wound healing. Cancer is a big, area of research in our lab. We have, 3 people just working on cancer right now. And then performance in extreme environments. So that could be resilience against hypoxia, resilience against extreme increase in oxygen or pressure. So these are the things that we're looking at. So we've broadened out into many different applications. My original application was very esoteric. Oxygen toxicity seizures are experienced in a pretty small subset of the environment. The limits are pushed with hyperbaric oxygen therapy. So oxygen toxicity is a limitation of hyperbaric oxygen therapy and will limit the therapeutic efficacy of oxygen toxicity or oxygen therapy, hyperbaric. And that's used for things like carbon monoxide poisoning. So you can really only go to about 3 atmospheres, 2.5 to 3 atmospheres of hyperbaric oxygen if you have carbon monoxide poisoning. If you could go to 5 or 6, if you were to put someone in ketosis, we have evidence that you could go to 5 and be okay and probably push the limits of safety. And then you could push, the carbon monoxide molecule off the hemoglobin molecule with enough oxygen. So, that's sort of the area that we're going into to make dieting for special ops safer and to decrease maybe the side effects associated with the high pressure oxygen over time. And then from the work that we did, on that project, we observed all these other benefits, and that expanded into a number of other projects that we assigned PhD students to or postdocs to, and they are just kind of running with it. And we're doing a lot of pharmacokinetic studies, a lot of safety studies on the racemic salts, and also we're comparing the R versus the racemic in various formulations. We probably got 20 different formulations that we're looking at. And the most important thing for me is to establish that they are not only efficacious but also safe. And I think safety is the number one priority. I've served on a lot of different panels to assess the safety of other types of ketone esters and salts. Mm-hmm. So I'm very familiar with the whole body of literature. Very familiar with Dr. Beach. He was one of the first people that I reached out to, actually one of the first ketone esters that we tested. But it wasn't effective against oxygen toxicity seizures because you also have to elevate acetoacetate in a certain ratio. And then that got me to kind of look into my own methods of development and looking at different esters from there.
So if you're listening to this and going, what the heck, racemic mixtures, hyperbaric stuff, we're getting pretty deep on this. And the reason this is so profound is that, in fact, I'm working on a new book about mitochondrial functions. I'll have to ask you a few questions off air about some nuances of ATP formation. But what's happening is we have something called the Krebs cycle inside our cells. And this is how your cell makes ATP and how it turns food into electrons. And you use those electrons to basically run your brain, run your cells, run your pumping systems, basically everything in your body. It's an incredibly complex thing, and it was only hypothesized and theorized in 1937. So in the last 80 or so years, we've come to understand what is really a little semiconductor inside our cells that has amazing properties, that it's affected by light, it's affected by temperature, it's affected by all sorts of environmental things. And we thought for years it only burned glucose, except if you were really, really sick. And it turns out that when it goes into its fat burning mode, ketosis, that essentially things that shouldn't be possible become possible. And I think you and I, Dominic, are not the first people to figure this out. Probably the first people to figure this out were thousands of years ago when they were fasting in caves, because after 4 days of fasting, your brain wakes up and you're like, wow, I feel amazing even though I've had no food. What's going on? What's going on there is what happens when your mitochondria have more power because they're using ketones, or at least have different power. And what you've done is you've kind of opened this giant maze, because you start out by looking at oxygen toxicity in special forces divers, like such a niche thing. And all of a sudden you're like, well, wait, Alzheimer's, ALS, Parkinson's, heart disease, diabetes. Like, is there any chronic disease that you don't think ketosis would help?
Yeah, that's a good question. Well, any kind of, from a neurological perspective, all neurological diseases are linked pathophysiologically to energy dysregulation, glucose dysregulation. So Alzheimer's disease, the gold standard for assessing someone with Alzheimer's dementia is a PET scan, right? A dim PET scan shows impaired glucose metabolism, and ketones can kind of fill that gap and help you restore brain energy metabolism. You generate more energy per oxygen molecule with ketones than you do with glucose. For example, the energetic efficiency of the mitochondria is increased. So the electron flow through the mitochondria to generate the hydrogen ion gradient that spins the ATP synthase is greater. So you have less reactive oxygen species produced when you generate ATP from ketones than you do with glucose. So Dr. Beach probably went into how it oxidizes Q, and then you have less superoxide anion production. And so that actually attracted me, because oxygen toxicity, the theories were that it was generating reactive oxygen species, and then that was contributing to the seizures. But it's really, the source of reactive oxygen species is the mitochondria. So if you enhance the bioenergetics of the cells by enhancing mitochondrial ATP production, then you don't make as much superoxide. So instead of throwing antioxidants, onto the brain and trying to prevent oxygen-free radicals during it, why don't we just enhance mitochondrial function, and then we could preserve the bioenergetic state of the cell in the face of the oxidative stress, which would be the high oxygen content. And that's why it works. I mean, it's and you could say that with a number of epilepsies. So the etiology of epilepsy is largely unknown for many cases, but nutritional ketosis and fasting work sort of independent of the etiology across all different seizure types by enhancing the bioenergetic function of the brain cells. And I think we're just beginning to understand sort of how that's working. And it's not one pathway, like drugs target one pathway. It's many different pathways sort of synergistically working together in synchrony to enhance and preserve brain homeostasis. And that's really important to understand. So, and that's metabolic physiology. And, it's important to distinguish, metabolic biochemistry from metabolic physiology, because a biochemist, which Dr. Veach is a biochemist and he's very knowledgeable, and he also has knowledge about physiology. But when you view it from a systems level, that's really important to understand sort of even the neural control of autonomic regulation. So how it's impacting our central nervous system, which are things we're looking at. We're studying the effects of ketones on anxiety. So just simply reducing anxiety, we find that it's like a whole nother variable. We see like a 30% reduction in anxiety. We just presented this work in Budapest last week.
Oh, wow.
And maybe the reduction in anxiety may be contributing to the delay in seizures that we're seeing. So we need to weed all this out. So there's many factors.
I've been giving some talks. In fact, I gave a talk at Jim Quick's Superhero U, like a kind of a brain hacking summit this weekend, and I'm talking about the bioenergetic effects of anxiety. And just it burns energy to focus on stuff that, like, a brain paying attention uses more electrons than a brain doing nothing or a brain at peace. So if you're burning more energy because you're worrying because you have bad programming and you're hungry, like, of course hunger makes anxiety worse. So if you have hunger, which is a problem with energy regulation, right? Like, there's not enough energy, that's why I have a hunger signal. Of course anxiety is going to be worse. So what do? Eat enough so that you're not hungry right when you're done eating would be a good way to start. Add more fat and get some ketones in there, and magically people, they feel less anxiety. But you actually quantified it, which is awesome. Like,
yeah, and when you achieve like a flow state, your brain energy and metabolism kind of goes down, or the crosstalk in your brain is kind of shut off, so when you achieve a flow state, you kind of dynamically activate the areas of the brain that you need for the specific task while kind of tuning out the others. And that's important to understand. And I think that being in a state of ketosis may help with that, and some drugs sort of help with that too, but nutritional ketosis is very effective, we think.
It's one of those things where if you look at mitochondria as being really important, ketosis, because it affects so many different parts of the mitochondria, is probably the biggest hack you can do to have better functioning mitochondria. Are you familiar with Dr. Schallenberger's work on ozone therapy?
No. I get that question a lot, though, because a lot of people ask about ozone therapy for cancer and for other things.
The similarity it has with ketosis is that it changes the NAD plus to NADH ratio by adding electrons directly from ozone. Like, there's a free electron in ozone. So you end up having a mitochondrial effect that is not dissimilar to the stuff that you're doing. But the reason I'm asking is he has found that about half of people, 46% of people, have early onset mitochondrial dysfunction before age 40. As measured with breathing oxygen, essentially looking at oxygen consumption rates. And okay, that's 1 in 2 of us has broken mitochondria and don't know it. But with the work you're doing around mitochondrial uses of ketones, well, just like you said before, you're reducing free radicals here, you can actually fix that. So I'm spending a lot of my energy working on how do we take people who don't know that their mitochondria are jacked and then getting them jacked in a muscle building jacked kind of way. Like my goal is, if you were to like take one of my cells out and look at it under a really fat microscope, you'd see like incredibly ripped and muscular mitochondria just like completely covering the thing. So like, I don't want to be a bodybuilder on the surface. I want to be like a cellular bodybuilder. So I'm focusing on like, what are the things I could do to grow better and stronger mitochondria? Given all of your ketone research, what are ketones going to do for someone listening to this who wants to have better functioning cellular energetics, like better mitochondria?
That's a good question, because they work through multiple mechanisms to enhance mitochondrial efficiency and also ultimately, from a more long-term perspective, mitochondrial biogenesis.
Yeah.
So ATP production is limited. And if we do like isolated mitochondria, there's a state 1, state 2, state 3, sort of, you can characterize how the ATP is being limited. And one of the states is reduced, TCA cycle intermediates. So you could have sort of reduced energy flow to the mitochondria to produce ATP. Now, ketones, being in a state of nutritional ketosis, is anaplerotic, right? So it helps generate the Krebs cycle or the TCA cycle intermediates. So succinate, malate, fumarate, alpha-ketoglutarate, succinate, all these are elevated like twofold, or even up to like threefold higher in the brain and in the serum in when you're in a state of nutritional ketosis.
Now, let's pause for a second. For people listening, the TCA cycle is the same as the Krebs cycle. This is how we make ATP in our cells.
And the Krebs cycle should really be called the Zaint-Georgi cycle, because it was a guy in Hungary who probably did most of the work on the cycle. So that's kind of an interesting history there. So generating Krebs cycle intermediates is one way. So increasing the bioenergetic molecules that create energy, right, is one way. Increasing oxygen flow to the mitochondria. The ketones enhance. One thing we're looking at in our wound model is vasodilation. So ketones seem to help couple the energetic state of the cell with oxygen and substrate flow. So it seems to enhance the vasodilate areas that need it most, which would be like an ischemic wound. In our aged model of ischemia, where the wounds do not regenerate efficiently, we can do a Doppler blood flow measurement and show a 30% increase in blood flow to the wound tissue, which has like a 90% deficiency in ATP production because it's not getting enough. So an increase, and that was shown years ago, showing like a 30% increase in brain blood flow or oxygen, brain blood flow with ketosis.
Now, what about like burn victims and people with injuries outside the brain? Is that still going to be really effective?
Yeah, we think so. Of all the things that we tested so far in our animal model of wound healing, the ketones are like one of the most effective things that we've seen. And what's interesting with all wound healing therapies are sort of tried topically, where you put the substance on the wound, where what we're doing is inducing acute and sustained nutritional ketosis with a supplement. So by altering metabolic physiology from a systems level, by lowering blood glucose, we know that if we give an acute bolus of exogenous ketones, blood glucose goes down. If you bring blood glucose down, by virtue of making your blood less sludgy, you will increase perfusion of the tissue. So it lowers blood glucose, it elevates blood ketone levels, it also elevates adenosine levels, which is a potent vasodilator. So it helps to perfuse the tissue. So just getting back to the mitochondria, it's anaplerotic, so you get more TCA cycle intermediates, you get more oxygen and substrate to the mitochondria. It enhances the bioenergetic sort of capacity, the electron flow through the mitochondria. It helps, obviously, to generate the reduced intermediates that drive the electron transport chain through the anaplerotic reactions. And then there's less reactive oxygen species production at the semi-ubiquinone site of the mitochondria. There's less superoxide. And making superoxide at the level of the mitochondria could be damaging, because that superoxide, if it goes to hydrogen peroxide in the presence of copper or ferric iron, you can generate reactive oxygen species through the Fenton reaction. And that can directly damage the mitochondrial membrane and decrease mitochondrial efficiency. You get electron leak, and then you just make less ATP. So we know, so that's 4 things right there. I mean, there's probably about 10 things in my head that I could talk about. TCA cycle intermediates, electron flow, more oxygen, less ROS production. So there's a lot of ways, even kind of taking a step back, the role it has on glucose homeostasis and all the other signaling pathways that could be activated, including AMP kinase, and that gets into a whole bunch of secondary reactions or signaling pathways that I could talk about. And importantly, too, I described to you sort of acute things that can enhance energy, mitochondrial production. And then we know that ketones are more than just an energy metabolite, right? They function as a histone deacetylase inhibitor, class 1 and 2. It was a science paper.
And so let's talk about the impact of that. Just people listening don't know what that is.
Okay, so if we're in a state of ketosis and we have beta-hydroxybutyrate elevated in our bloodstream, that's impacting gene expression in a way that's altering our physiology over time. So, and it's decreasing oxidative stress. It's conferring protection against aging and oxidative stress over time by increasing the activity of things like superoxide dismutase, by activating a whole genetic program that's kind of parallels calorie restriction, right? So we know all the benefits of calorie restriction have been sort of really talked about in the literature. It's easy to find that literature, like on PubMed. One of our colleagues, actually, and collaborators from Yale, Deep Dixit, contacted me and did a lot of work with calorie restriction and fasting. And he said, we think beta hydroxybutyrate may be like the metabolite that's actually conferring all these benefits to fasting.
Wow.
So he contacted me, and we formulated a diet. We just basically took the standard diet and formulated a diet with our ketone ester. And we collaborated with him, and he ran a study, and he found that elevating beta hydroxybutyrate suppresses the NLRP3 inflammasome. So that inflammasome is linked to a whole host. It's like the hub inflammasome that kicks on the inflammatory cytokines that are associated with many age-related chronic diseases. And it was published in Nature Medicine. And it's like that's sort of like the gold standard of scientific publications.
It's kind of interesting. Bulletproof intermittent fasting, where you have bulletproof coffee made with brain octane in the morning at the end of the fast, while you sleep. Brain octane raises BHB more than MCT oil and more than coconut oil. I've been doing that every morning for like more than 7 years now, and it's completely changed my life. And I believe it's a mitochondrial energetics thing. I also think that one of the reasons caloric restriction works, and by the way, I have no scientific basis for this other than I'm a systems guy and I'm a biohacker, right? Mitochondria, when they're burning sugar, and when they're burning anything, they do produce free radicals. So if you eat less food, you make less energy, you have less free radicals, you get less damage. So therefore that could confer some of the benefits, but they come at substantial cost as well. Like you're cold and tired and having less energy sucks. I like to be like exploding with energy, but I don't want to die when I'm 50. So like, how do you manage to have more energy without incurring the damage from the energy? And like, that's my quest right now. And I think ketosis is like at the core of it.
And I think, and your brain octane has been something that has been a staple in kind of what I do. The C8 is a pretty potent, the more, we're studying, these synthetic expensive ketones and ketone esters and ketone salts. But the more that we look at what's in brain octane, C8, the more impressed I get. It's a little bit biased because in rats, they have such fast hepatic metabolism that you can get blood ketone levels. It mimics, you get the same effect as a ketone ester, as you do with C8. So you can easily
in
get
rats, but not in people, right?
Rats, and if you gavage. So the C8 is interesting because it goes directly it actually perfuses the liver with the oil when you drink it instead of it being packaged into chylomicrons and going through the lymphatics. And you get obligate oxidation of the fatty acids exclusively in the liver, and not much C8 actually escapes the liver if you keep the dosages even in the moderate to high range, and it almost completely gets oxidized in a healthy liver to beta hydroxybutyrate. And it doesn't get, so the MCTs and C8 especially they're not esterified. It's poorly esterified. It doesn't get transported by CPT1, the carrier, and it can be independently go right into the mitochondria, and it doesn't need a lot of carriers. We also found another benefit of MCTs that I didn't know until I did the metabolomics is that they readily cross the blood-brain barrier. So not only do 90% of it gets kind of generated, 80 to 90% seem to be generated into beta hydroxybutyrate. And then what does get into circulation can readily cross the blood-brain barrier. I thought fatty acids didn't, but short chain and medium chain do. And we, when we took the hippocampus out and did the metabolomics on it, we found pretty high levels of C8 and C10. We did MCT and C8. And so the MCTs are very versatile ketogenic compounds that can be, they taste good. You can formulate so many great carriers?
Some of them, though, like this is something that I've helped to push the MCT thing out there, but there are companies selling some that contain C6, which is like a really strong gastric irritant. So you end up, frankly, shitting out the rest of your MCTs when you have that. And then C12, which is good for you. It's not really, like, it doesn't go to BHB. It acts like a long chain fat, but it's legal to call it an MCT. So you're seeing like coconut oil companies pretend that there's a 60% MCT when it's really 15% of the MCTs that actually work. So I'm seeing like a lot of kind of slimy marketing activity. I'm sure you are too. And MCTs, at least the ones that work, they're just like step one of ways you can increase ketones. And you've got like two other ways of increasing ketones that I wanted to pick your brain about here, because I synthesized my own esters 3 years ago, but it was $30,000 a kilo. I'm like, I can't even afford to take these myself, but I took like 2 samples and it worked. And then there's salts too. So,
what ester was it that you synthesized? Do you remember?
I would have to look in my notes, but I worked with a third-party lab, and it came in a little vial, and it was insured when we shipped it because it was so darn expensive. Geez, I'd actually probably have to open up an old email to find it.
Actually, yeah, the first time, this goes back to 2009, that we sort of acquired different ketone esters and to test which ones were going to be effective against oxygen toxicity. My original proposal to the Office of Navy Research was to feed it for 1 week, because I was convinced that you needed to keto adapt the animal, or at least get levels, the tissue saturated, the brain. And I only had enough to really give 1 dose, and I did it 30 minutes prior to a super deep oxygen dive. And I was like, well, I don't have enough to really run the study that I want to do, but I have enough to give 1 dose prior to, and we gave 1 dose prior to the dive, and we saw, in every single animal we dove, we saw an astronomical increase in the latency to seizure. And we published that, and it was like a 575% increase in the latency to seizure. And that pretty much outdoes all anticonvulsants that, the military has looked at for this. And we still have not gone back and did a study where we feed it continuously. We're doing sort of some work now, but I was basically figuring, if it's this effective, just giving 1 dose in an animal that's eating a high carbohydrate diet prior to the dive, like really that's what the military wants. They don't want to like have to feed something for a week. They want to give like 1 shot and then, hit your mission. So I basically, we have long-term chronic feeding studies for other disorders, but when it comes to oxygen toxicity, we're really just looking at kind of, a single dose prior to the mission. But we screened out, the different types of exogenous ketones in that project in the beginning and identified the one that was most effective, and just been really focusing on that. And getting that synthesized was a project. Yeah, it was a lot of back and forth with the chemist. So.
Yeah, I don't think that problem is solved, but manufacturing that stuff is just complex. And people listening like, oh, big companies can just make whatever they want. This is one of those game-changing supplements that has the possibility to transform a lot of athletic performance and a lot of just like, if you want to feel young when you're old, have mitochondria that work. Like the average 70-year-old has half the mitochondrial function of someone who's 30. And it's one of the reasons it sucks to be old. So like, let's get ketones in older people, and magically, like they'll get up and dance. Like it's that big of a difference. And so I'm very hopeful, and I don't think Bulletproof is big enough to fund the kind of manufacturing research that's required to do this. Like it is a giant project, but I'm working on it, and I'm supporting everyone I know in every way I can, including just by talking about how important this is. But let's talk about salts before we get into esters. So there's like a hierarchy of ketones, and the first thing you do is you go on a low carb, high specific fat diet, right? That alone can put you in ketosis, Atkins, stuff like that. And then the next thing you can do is add brain octane, which is basically the bulletproof diet, which gets you in ketosis, especially in the morning. Do this bulletproof intermittent fasting, no carbs in the morning, no protein in the morning, extra fat in the form of brain octane, as much as you can take without filling your pants, right? And you do that, you're like, okay, something different just happened. I can get my levels up to 0.8 or 1.0 in my blood just from brain octane. Nutritional ketosis starts at about 0.8. And then from there, though, there's ketone salts. Talk about what the salts are, the 4 kinds of salts, what are the pros? What are the cons? And if you could address what Dr. Veach in his interview was like, well, some of these based on whether they're bioidentical isomers, this is called, that basically mirror image molecules of each other, whether that's an issue or not. So kind of walk me through the salt story.
Mm-hmm. Yeah, it's important to address the racemic issue because it can create a lot of confusion. So I'd like to address, danger, like, are they dangerous and are they efficacious? And I think that's kind of what I'll address kind of up front.
Yeah.
And it's important when we approach this question, it's important to distinguish and separate someone's opinion from the scientific fact and what has been published and observed in the literature. So when I listened to about like half of the podcast, or I was driving, so I don't remember all the.
I get it.
I doubt that he referenced any papers when he said ketone salts were dangerous, because I've sat on many boards, and it was actually in talking with him, it was a concern that I had. Because we were testing a number of different compounds, and the ones that were most effective for my applications tend to be the ones that were racemic.
Explain what racemic is for people listening who don't have the biochemistry background.
Yeah, so there's different, probably be good to look up the word enantiomer, and it's a stereo sort of image of when you think of your hands as sort of the mirror image of it. So the molecules in your body, most drugs that we consume actually are racemic. So the way that if you go back to general chemistry, when you had when you did all the stereochemistry of the molecules and you were studying that, if your listeners out there had general chemistry, you probably remember you probably came across the word racemic. So if you pull out your general chem book, you can look up the definition of that. But essentially, there are molecules in your body, amino acids, lactate, for example, is L-lactate would be sort of the physiological enantiomer, and D-beta-hydroxybutyrate would be the physiological enantiomer, and the opposite enantiomer has a configuration that would mirror it, sort of like two hands coming together. And depending on the specific metabolic pathways, the body will reject it or consume it. Or some may have a weak interaction at the receptor if it's a drug, or a stronger interaction with the receptor. So one thing I got interested, I studied lactate, the effects of lactate like ketones. So lactate's an alternative energy substrate. Before I got into oxygen toxicity, I was looking at hypoxia. And lactate is actually a very effective fuel. When you exercise, your body produces a lot of lactate, and the lactate actually goes to your brain and functions like ketones. It's like an alternative energy substrate. And through the Cori cycle, it gets recycled back to glucose. But I was interested in using lactate to protect the brain from hypoxia and ischemia. And alpha-L polylactate is something that I was interested in. As a cyclist, it's in a product called Cytomax, and I was interested in developing different forms of lactate. And I saw that racemic lactate was being used as ringer solution. So any ER doctor, any medical doc out in the field, when you're shot, when you lose blood, when you're in the emergency room, they fill you up with Ringer's lactate, which is racemic lactate. So, according to Dr. Veach, that would be dangerous. That would kill you. But it saved millions of people's lives. So they always use racemic lactate for that.
So what this, for people who didn't take general chemistry, imagine that like every molecule in your body likes left-handed things, but sometimes you have right-handed things. Like it's still a hand, but it's not the same hand. And sometimes it'll fit into kind of the glove-like receptors in the body. And that's a gross simplification. But the idea there is that they're almost the same, but they're just mirror images. And so if we're left to our own devices biologically, we typically make the L form, right? But the opposite form we can make in the labs. And when we use it as a food supplement or as a drug, it does all sorts of cool stuff in the body, including sometimes kill you. But quite often it's useful. And Dr. Veach's concern when he was on the show was that if you're going to be magically raising ketones, which frankly give you superpowers, like you get more energy than you had before, like Captain America kind of stuff. Well, you want to make sure that you're not doing something that's going to give you cancer or increase oxidative stress or otherwise shorten your life or mess you up. And so Dr. Veach had a pretty good concern here, and what you're saying now is kind of the counterpoint that says, well, okay, here's all these times when we use the wrong hand in our bodies and it completely saves lives and it completely works, which is a pretty powerful argument, I'd say. Dom, you've got a point there.
I mean, we don't progress, as a species unless we kind of move forward on truth, right? And from my perspective as a scientist, the truth should be in the science. So if there's evidence out there, that racemic salts are dangerous, or that Ringer's lactate is dangerous, it saved millions of lives in the emergency room and on the field, that using these racemic compounds are dangerous. There's just no evidence. And then one could, ask the question, is one optimal over the other? And I know, lots of work was done early on Ringer's lactate, and they compared L versus racemic, and there was no change in metabolic state on its buffering capacity and its capacity to save people's lives. So I assume the same thing would apply for ketones, but we're interested in studying that, and that's why we're studying that now.
But there are people selling it right now, maybe who haven't tested it. What do you think about that?
Yeah, so I was a little bit concerned until I went, at about the same time that marketers started, commercializing ketone salts, was about the same time that I was on various, panels, expert panels to really, just spend weeks going through the literature to see if these things could have any potential for danger. And I just couldn't find any. And I kind of wanted to find something because I thought marketers were kind of jumping the gun in certain, but I couldn't find evidence to suggest that they were dangerous or less efficacious. And to that argument, you see Ringer's lactate has been used for decades, and that the DL, the racemic beta hydroxybutyrate, has been used for decades to treat a disorder called MAD. And MAD stands for multiple acyl-CoA dehydrogenase deficiency, MAD. And the kids with this, the ketogenic diet doesn't work for them, but exogenous ketones do. And they're given very high doses of pure sodium beta hydroxybutyrate to stay alive. And this has been published. There's lots of publications on it. There's a publication in The Lancet, which has like an impact factor of 45, showing the safety and efficacy of sodium racemic beta hydroxybutyrate given in massive quantities to kids over years to manage their disorder. So there's, you find, and that's just one example. I was going through a list of emails this morning with parents that have kids that have glucose transporter type 1 deficiency syndrome. And for reasons, a lot of these kids tend to be picky with their diet, and they found these ketone salts on the market, and they ordered them. And I've gotten emails from from parents that said when their child drank these ketone salts, they literally just woke up. It's like they came out of a coma, and they're energetic, they're lucid, they're active for hours until it starts to wear off, and the ketones come back down. And then they dose them up again. And you have like 4 companies out there, making them. And they're all, they have sodium beta hydroxybutyrate in it, but there's also potassium and calcium and magnesium. And I know Dr. Veach kind of talked about the sodium being an issue, but sodium's, has not been an issue for all the applications that it's been used for. And the products on the market, they spread the beta hydroxybutyrate across a number of different monovalent and divalent cations. And that's really important. And that's really what I want to do, is formulate something that has, at least a dozen or more different carriers of beta hydroxybutyrate. You can use a list of amino acids, calcium, sodium, potassium, magnesium. I have magnesium beta hydroxybutyrate right here. I was just testing.
And actually played with it too.
And I just took pure magnesium beta hydroxybutyrate and can get up to about 0.5 millimolar, by itself with no GI issues with this particular magnesium product. So, you can envision, getting about 6 different either amino acids or monovalent, divalent cations in formulating something that tastes good. That's really important too, because some of them don't taste good. They need to be buffered. So it's not, when we talk about ketone salts, a lot of people just think sodium. And sodium, you can have I have a sodium-free ketone salt that I've been testing that has no sodium in it, and it tastes really good and gets me up in the 2 millimolar range. So the sodium is not really an issue. I personally don't have an issue with sodium. When you're on a ketogenic diet, your insulin tends to be a little bit low, so you excrete a little more sodium. So if your RDA is like 2,500 milligrams per day, you should probably be taking more like 3,500 or 4,000. And that would allow for a pretty significant amount of sodium beta hydroxybutyrate, to get your ketone levels up without any problems at all.
I actually target 4 to 6 grams of sodium in my diet per day. And there's abundant evidence that that is safe. In fact, lowering it to the current limits that the FDA recommends actually increases heart attack risk in studies. So like sodium, if you're a salt sensitive hypertensive, like you basically get high blood pressure from salt, 2% of people are like that. Restrict sodium. For the rest of you, if you're stressed, you need more salt. Like, go for it. It's fine. And if you take the salt with ketones, okay, that's totally cool.
That's funny. That's about what I get per day too. I look at my sodium load. For a while, when I was testing just the sodium, I was getting about 10 grams per day, and I didn't have a problem with that. And
I've done 10 a day, and it helped me recover. I don't need that much now, but absolutely, it's just fine.
Especially, if you're out, we live in Florida, so it's like if you're sweating, if you're outside working out, I think 10 grams a day may actually be optimal. If your energy levels are low, just take in some sodium with ketones, and it'll wake you up and energize you.
When I was in South America, I was doing some high altitude mountaineering at Mount Cotopaxi in Ecuador. And I was literally adding a teaspoon or 2 of sea salt to my water bottle and some amino acids. And all my other climbers were like, what the hell are you doing? And I'm like, I'm feeling good is what I'm doing. Like, you need the sodium. And it's one of those things where if you're getting too much salt, your body will tell you very abundantly. Yep. So we just pissed off half the physicians in the country, but sorry guys, read your science.
Well, it happens. When people talk about the dangers of a high sodium diet, we're administering the sodium in processed food with refined carbohydrates, which spike your insulin. And the insulin makes you retain the sodium. So that sodium retention, it's really not about sodium intake, it's about sodium retention. And insulin causes you, to massively retain sodium. Just look at someone who's done like a pre-contest sort of physique, dieting for a bodybuilding show or something. And they go, from calorie restriction or low carb to high carb. And just going high carb, that insulin spike with sodium, they blow up so fast. It's the insulin really that's causing the sodium retention. And your insulin levels are suppressed, on the Bulletproof diet, on the ketogenic diet, low carb. And when your ketones are elevated, your, hepatic ketogenesis is driven by suppression of the hormone insulin. So you are excreting more sodium just by virtue of keeping your insulin low, driving beta oxidation in the liver. So that whole process is just excreting more sodium. So it makes sense to get more into your diet. And that's good because I like the taste of salty food.
I actually travel with a little vial of salt. I'm like, some restaurants don't put salt out. I'm like, what's wrong with you? So I just put it on myself. Yeah. Now, I have a question for you about mitochondrial physiology here. And so I'm writing my next book. It's like, how do you hack your mitochondria? And I'm really getting into it. And one of the things that really matters is the size of the mitochondria, because if you basically move molecules apart a little bit, it takes a lot longer as a percentage for these little electrons to move. And one of the concerns about using ketone salts is that if you're using the racemic form, that it's going to reduce mitochondrial NAD, which causes oxidation of Q, which basically increases the redox span between these two parts of the electron transport system. Now, if you're listening to that, you're going, what the hell did Dave just say? What I'm saying is, if you imagine that you have to take this little packet, an electron, you have to move it from step 1 to step 2 to step 3. If you move the steps further apart, it's going to take a lot longer. And if you have to do it 100,000 times, all of a sudden that increase in distance becomes really meaningful. Are you concerned about that?
No, because I understand sort of the metabolic biochemistry of it. But what our lab sort of does like this top-down approach where we feed the ketogenic diet or we feed ketone supplementation, and then we go back and look at the brain tissue, the skeletal muscle. We do metabolomics. What we see is all the evidence points to enhanced, more robust mitochondrial function.
Okay.
So, and we can look at, using electron microscopy, we can count the number of mitochondria, and now we can also look at the morphological structure of the mitochondria. And I just got back from Budapest, and I was talking with an MD PhD over there that has hundreds of brain tumors, and he said, even in low magnification, when you look at a tumor, the mitochondria, the inner mitochondrial membrane, is kind of all mucked up. I mean, It's even low magnification, you could see total crystallosis. You can see malformed mitochondria. Some of them are enlarged, some of them are small, some of them are split in two. A lot of times you see them forming like little circular structures, which means they're stressed. And then in healthy tissue, we just see like a very sort of dynamic dispersal of mitochondria, and also the ability to mitochondria for them to migrate, for example, in brain tissue to the synapses, where there's sort of robust sort of bioenergetic processes happening. A lot of electrochemical gradients being shifted from bioreaction potentials and things like that. And the mitochondria are sort of transported through the cell in different ways. And they all, in healthy cells, what you see is they all sort of have like the same kind of uniform, sort of circular football kind of shape structures. And then in disease tissue or disease model, you just see like a range of different mitochondrial, sizes and shapes and morphology. But with the ketogenic diet, or animals or humans are fat adapted, you do see more mitochondria, and people have reported this, and we have a lot of tissues to look at. Sort of just the architecture of the mitochondria are more consistent with healthy mitochondria. They have a more intact cristae, and that's really where all the action's happening, right? The inner mitochondrial membrane, right at the level of the cristae, and that's really the indication of the bioenergetic capacity of the mitochondria, the health of the inner mitochondrial membrane, and that's held together by different substances, and probably the most important being cardiolipin. And in cancer, what you find kind of a shift towards more immature cardiolipin, so the inner mitochondrial membrane doesn't stay together. And with the ketogenic diet and with healthy mitochondria, you see a shift towards more mature cardiolipin that sort of plays a big role in mitochondrial health and mitochondrial efficiency by preserving the integrity of the inner mitochondrial membrane. And that's really a function of ketones, the ketogenic diet, a healthy kind of complement of essential fatty acids, phosphatidylcholine, phosphatidylserine, things like that can enhance healthy mitochondria over time.
It's such a complex topic, and that's why I decided that my next book needed to be about mitochondria, because I've been following the work of guys like Nick Lane, and I just realized everything I've ever done that really made a huge difference, including back in the day, I used to shine an infrared LED, one of the first LEDs you could get, on my head where there's the most mitochondria, and light affects mitochondria dramatically. So I've been doing a lot of light-based interventions for mitochondria for more than 10 years, and I see what happens when I mix ketones, which is like our main topic, with other things like cryotherapy that actually makes mitochondria work better because they get a little smaller when they're colder, so electrons move faster with ketones. So you stack ketones on almost anything that makes you perform better, and you're like, oh, like something magic happened. So I'm excited to explain this in a way that is less sciencey, but man, it's pushing my limits of being able to simplify because it is such a complex topic.
Well, these books won't simplify it, but these are excellent books that, kind of...
So hold those still, I'll read them. Mitochondria is one of them. Who wrote that one?
Yeah, there's a number of authors, so there's editors, basically. So there's like probably 24 authors. To get into the level of detail that you need to in here, you have like a whole bunch of authors.
Okay, got it. So those are basically medical textbook level things.
Scheffler is the author of this book.
I may have that one. I don't have the mitochondrial cancer book. We'll put the names of these for there's probably 20% of people listening to this right now are in the medical or healing fields. It's amazing the number of emails I get from people who are like, I'm at this big hospital, I'm doing this kind of research, and they listen because we're willing to go deep enough. And there's tons of people who aren't in medicine at all who are just like, how is it really true that you can do this? And of all the guys I've ever talked to, man, you're like one of the top ketone experts out there. And ketones are a superstar when it comes to just performing at a new level. I wouldn't be sitting here today without ketones for really more than a decade because my brain was such junk without them, to be perfectly honest.
Yeah, I don't think I could do, I just flew in overnight last night, and I don't know, I've been awake for, I'm not even counting the hours now. I don't really think I could function in the capacity that I do with the productivity that I have been doing to get tenure recently at my university.
Congratulations, by the way.
Thank you. It took a lot of overnights, working on grants and publications and things like that. And I don't think I could do it if I didn't follow a nutritional program that allowed me to have the resilience and just stay up night after night and work on things. And I'm in a pharmacology department, so I'm really supposed to be working on drug research. But when the drugs didn't work for the type of seizures I was looking at, and I found out that the ketogenic diet through the Charlie Foundation was being used at Johns Hopkins for hundreds and thousands of kids, and that it worked about 60 to 70% of the time, even when drug failed. I realized it was something I had to look into. And then when I realized that you can actually create exogenous ketones to mimic that, then I got really interested, because that's kind of like the combination of nutrition and pharmacology together. So it was sort of accepted doing, high fat diet research, for neurological diseases in a pharmacology department is a little strange. But if you're developing, a dozen or more different types of synthetic agents that can mimic that neuroprotective, state of nutritional ketosis, that's something you could build a career off of. And I realize I'm very fortunate that I just stumbled across Dr. Veach and Henry Brunengraber. He was at Case Western. He helped me out a lot. Sammy Hashem, Theodore Van Italy. He's 96 or 95 now. Wow. Ketogenic diet guy. These are the pioneers in the field, and these are super smart guys who really understood metabolic physiology. And Dr. Cahill, so George Cahill at Harvard Medical School, did the experiments fasting, subjects for 40 days. And he passed away a few years ago. But, when I was in my postdoc, I was calling him up, all the time. I was calling all these guys up. I was probably really annoying to them. They always had questions,
They probably loved it. I don't think you're annoying at all. What it's like when someone who really cares about something calls up. I get calls like that sometimes. And you're like, of course you want to talk about it because it's your life's work, right?
Yeah. They had great stories to tell, a lot of stories. But yeah, they were really kind of inspired that a younger guy was kind of picking this research up, and they inspired me just because of their background, that this was something real that I needed to get into more and study for the applications that I was looking at.
From a personal perspective, about 3 months ago, I got food poisoning and I threw up so hard I passed out and I smacked my head on the tile and got a concussion. I actually seized for a few seconds. It helps to be married to an emergency room doctor. But I can tell you with absolute certainty that I used every possible way of raising ketosis I had on hand, which was basically brain octane oil. But I absolutely did that, and I took a bunch of mitochondrial energizers. I manufacture a bunch of them, and I pretty much have all of them on hand because I'm always hacking my mitochondria. I use red lights and other things like that, infrared, and recovered very quickly, didn't get any long-term damage. But even with that, for about a week, I was kind of a zombie, where my working memory was shot. I couldn't remember words. I swore a lot, all kinds of weird stuff. I think if I hadn't had ketones present in my body during that time, I would probably still be swearing. And I would not have recovered at the rate that I did. And I did neurofeedback and a bunch of other stuff. So I came back actually remarkably quickly. But thanks for pointing out the things you pointed out in your work. Because of that, I knew what to do. And that's not the sort of thing you Google for. I also have hyperbaric oxygen downstairs. We talked about that earlier. I'm lucky to have a human hacking lab. But I look forward to the day when that knowledge is just common sense, and when a kid takes a hit on a football field, which happens tens of thousands of times a year, that we actually put them on ketones and we put them in hyperbaric and we do all those things, because that'll actually change enormous numbers of lives. I mean, just huge lives.
Yeah, I get emails from parents and their kid, their high school or even Pee Wee football, they've gotten a concussion and they ask, what's the best way to get my kid into nutritional ketosis? So I'll kind of give them.
Keep answering that.
Yeah, I get a lot of questions. So, and people ask, are ketones, it sounds like they're a panacea for everything. Soon we'll be saying they cure the common cold. And when I think about it,
they do
3 colds a year, and it's been about 7 years since I've been on a ketogenic diet, and I've only had one cold that I can remember, 1 or 2 maybe. And it was just like little sniffles, so maybe we get less colds as we age, but I went from getting 2 or 3 a year to 1 or 2 that I can remember over the last 6 or 7 years. So that's really strange for me. And that's through a lot of stress. And usually stress always triggered, because right around finals time when I was in college, I would always get a cold.
I'll sound like the first chapter in the mitochondria book, but like everything your body does is based on mitochondria, other than your red blood cells that don't seem to have them. But if you can do anything that just even gives you a 10% boost in the way they function, it's 10% more of everything, including immune function, including everything in the body. So I kind of look at that as the ultimate hack for the body, where of course your immune system is going to work better, because anytime a cell needs to respond to an invader, it's got more energy. If you have more energy, you're going to do a better job. And I think that works at the very micro level and at the very macro level. And like, I've used ketones to build my company, to write my books, to be a better parent. And you could use sugar as well. It just doesn't work as well. And so I don't. I'm same way. I'm not sick like I used to be.
Yeah, ketones were kind of an easy sell for me, because I was a patch clamp electrophysiologist in my PhD. That's what I did. So
wow.
With electrophysiology, with patch clamping, and I did something called a whole cell perforated patch, where you take a glass electrode and you put it on a neuron, and you don't dialyze the cell, but you seal onto it, and then you have nystatin in the pipette, and it pokes little holes in the membrane, and then you gain electrical access, right? And then you get the action potentials and the membrane potential. And it was very clear to me from even early on in my doctoral work that we're just like a bunch of batteries, right? And we have these bioenergetic potentials. And that was completely driven by sort of ATP, ran the pumps that created the membrane potential. And when I would de-energize the cell, either by cutting off the substrate or cutting off the oxygen, which my background is looking at hypoxia, it was, that's when life ceases to exist, right? I mean, that's when you shut off the energy pathways, and you can't maintain that membrane potential. And that membrane potential really is sort of like the definition of life. It drives all the biological processes that we call life. So preserving the ATP levels and the bioenergetic state of the cell with ketones, I mean, it just made a lot of sense to me. And that's when I first started studying lactate and then other alternative energy substrates. And I came across ketones, and it seemed such a versatile thing. There was ways to do it, through fasting with the ketogenic diet. And I discovered, other ways to enhance ketone production. And it was really like a super fuel. If you're at the gas station and you have 4 choices to choose from, it's like the super high octane stuff. So I wanted to study that. And so it was a pretty easy sell for me, especially since all these other therapies, all these other disorders will respond to this type of metabolic therapy. It's a very powerful metabolic therapy that has so many different applications. And if you look at my CV or what I'm doing, it looks like I'm all over the place, but that's just because, nutritional ketosis or therapeutic ketosis can be applied to so many different pathologies.
It takes you there. If you follow the ketones, you are going to realize that they're responsible for just about everything we do, because they have more energy in them than sugar does. And so more energy means doing more, right?
And fat does too. So
fair point. Yeah, fat, which drives ketones, right?
I always like to preface it by saying being fat and keto adapted. So
there you go. That's a good point. Now we're over a little bit on the length of the show, and I know you've just got off an airplane from Budapest, and I appreciate you making time to be on Bulletproof Radio. I've got one more question for you that I'm sure you don't remember your answer for because it was a couple years ago, last time you were on the show. If someone came up to you tomorrow and said, I want to kick ass at everything I do, I want to be better at everything I do, what are the 3 most important things I should know? What would you tell them?
Get enough sleep, right? So, yeah, I mean, the most important thing is just to follow what you're passionate about. Because I think even if you have you probably run into people who follow a crappy diet and don't get any sleep. But if they're really driven from a gut level and very passionate about what they're doing, and if what they're doing is not self-serving and it provides a service for someone else, then they become more intrinsically motivated to work harder and be more successful. So I would say, kind of sounds cliche, but follow your heart, or just follow really what you're passionate about. And if you follow nutritional ketosis and, proper sleep and get sunshine and fresh air and exercise on top of that, then you're really going to kick ass.
Love it.
You got to, focus on what you're passionate about and just don't have any fears and go for it.
I knew you were going to say ketones in there and you didn't.
I said diet, yeah. So
there you go.
Fortify that passion with nutrition and sleep and everything else, nutrition that involves ketones. And
there you go. I love it. Dominic D'Agostino, where can people find out more about your work? Are you going to write a book for all of us or at some point or something? Or what's the deal there?
Yeah, I'm thinking about it. But right now, I think what's important for me is to build a big foundation of research that the book will be about. So I'm kind of really deep into doing that kind of research. We have a lot of research projects that are going full steam right now. And in the process, I'm writing a lot of manuscripts and reviews and book chapters and things like that will ultimately become part of probably a book in the future. So I'm entertaining that thought. But ketonutrition.org is a website that I maintain and basically just put a bunch of links up there. And Metabolic Optimization is also a website that I started and did a few podcasts. And intend to have more people on that show.
Beautiful.
So those 2 links right there.
Thank you. I look forward to having you back on Bulletproof Radio, maybe in another 6 or 8 months. It's always a pleasure.
Thanks for having me, Dave.
Don't miss out. To keep getting great videos like this that help you kick more ass at life, subscribe to the Bulletproof YouTube channel at bulletproofexec.com/YouTube. Thanks for watching and stay bulletproof.